Hello friends, welcome back to my channel, and today in this video we are going to study about TCR and signaling. We already learned about B cell signaling and BCR, that is an antibody molecule, and the associated co-receptors. Similarly, we're going to study about TCR and its signaling pathway, which we already overviewed. So today we'll dwell little deep to understand the TCR. Now TCR, if you remember, in TCR we have CD4 or CD8, and because these CD4 and CD8 and this alpha beta is the main TCR receptor to which any antigen binds, because they have a very less cytoplasmic domain, so they need a long cytoplasmic domain so that the signaling pathway or the signal transduction can be proceed. So they have this CD3, which we already learned. Now there are two types of T cell receptors. The majority are of alpha beta type, but in some mucosal cell we have gamma delta also. Now the TCR proteins are the members of immunoglobulin superfamily. The C alpha domain of TCR differs from most immunoglobulin domain in that it possesses only single sheet rather than a pair. If you remember, in BCR we have the antibody, and we already learned about the structure of the antibody, that it remains in a pair form. Clear? So this is different from BCR. Now the transmembrane domain of TCR and the chains are unusual in that they contain positively charged amino acid. This we can understand better in the next slide. So now T cell signal transduction, it includes CD3, as we all know. Now CD3 is made up of three dimers. Either it will be of delta epsilon or gamma epsilon or zeta n type. So it is of three types, as you can see here also in this diagram. This is one type, this is another type, and this is the third type. Now the transmembrane region of these co-receptors, these are negative, consist of negative amino acids, because the main TCR, its transmembrane segment contains positively charged amino acid. So this positive can easily interact with negative to form what kind of bond? That you should let me know in the comment section. That whenever any amino acids are positively charged and in nearby any negatively charged amino acids are present, that what kind of bond they can form. Now coming to the cytoplasmic tail, if we see this blue segments, these are known as ITAM. What is ITAM? ITAM means this is immunoglobulin tyrosine activated motifs. We already learned that in cell signaling, here the phosphorylation of tyrosine takes place, that helps to activate this cytoplasmic tail and make the site for the attachment of any adapter molecules. Now these adapter molecules acts as a docking site for other signaling molecules. So this we learned already. Now this is the same thing. CD3 dimer contains negatively charged, that is there in this figure. Now you have to tell me why this difference in charged amino acid is necessary. Now if you talk about co-receptors, then we have CD4 and CD8. CD4 is present in T helper cell, CD8 is present in T cytotoxic cell. Now CD4, it is of 55 kilodalton, it's monomeric membrane glycoprotein, 4 extracellular immunoglobulin-like domains are present, hydrophobic transmembrane region are present, long cytoplasmic tails present. So as you can say, this is CD4, the figure of CD4. So this is extracellular, this is the transmembrane segment, and this one is the cytoplasmic segment. Now if you see CD8, it is quite different from CD4. It consists of this heterodimer alpha and beta. This is the extracellular part, this is the transmembrane plant, this is the cytoplasmic region. Now the co-engagement of the single MHC molecule by both the TCR. So the co-engagement of the single MHC molecule by both TCR and its CD4 and CD8 co-receptor enhances the avidity of T cell binding to its target. What do we mean by avidity? Check my earlier videos. You will get to know what is the meaning of avidity. Now coming to its signal transduction pathway, it is same like B cell signaling, only you can find some maybe names different. For example, one pathway is this again, calcium pathway, that is calmodulin and calcineurin pathway. Another pathway is DAG, which activates nuclear factor kappa beta, that is PKC theta. Another pathway is the RAS MAP kinase pathway, which activates AP1 and responsible for the expression of the gene. Apart from that, cytoskeleton reorganization also happens, and this is the anti-apoptotic pathway. So this we already learned in the B cell signaling pathway in detail. So it is same for T cell signaling as well. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.